Like they do a statistical test, but it's like it's there or it's not there.
Yep. Yep.
And and I think those are the kinds of experiments we should be striving for. We can't always do those, especially when we're dealing in, diverse genetics, when we're dealing with human subjects. Like stats have their place, but if you're going to use statistics, by God, use them correctly, please.
This is Audio Immunity, a podcast about our body's never-ending fight with the outside world.
Hi, everyone. This is Audio Immunity, episode 38, brought to you today by Leaky Guts. Is your immune system too uptight? Are you waking up to find that you have low tolerance to, well, everything? Try Leaky Guts. But wait, are you more than a few days old? Too bad. Should have tried Leaky Guts. We're recording on April 8th, 2025, and we're happy to be with you. Thanks for tuning in. Coming to you from up and down the East Coast, I'm Matt Woodruff, joined once again today by Kevin Bonham.
Hello, hello.
And Kevin, we have spent the last, 50 minutes trying
Yeah,
to get our normal recording apparatus, Riverside FM, to work, to do anything for you and I to see each other, communicate with each other in any
let's be
meaningful way.
clear. We were able to either see each other or hear each other or record, just
Mhm.
not all three of those things at the same time.
Right. Yeah. So the result of all of that is that we have decided to just do it live. So we're coming to you today on Zoom, and we are doing what those in the biz of which we are not a part called a one shot. We are trying to do it live, one shot through this whole thing, and so you will be with us through the ups and downs, the misspoken words, the lost trains of thought that generally are involved in our scientific discussions. I would say that Kevin often masterfully edits out.
Yeah, I mean, I'm gonna still try to edit it, but maybe just not quite as hard
Yeah,
as I normally would. So
yeah.
yeah, good luck to us all. Good
Yeah,
luck to us all in this.
fingers crossed. So we are not going to spend a ton of time jumping into this paper because I think that we have already wasted most of our recording time this
That's
morning.
correct.
So let's talk about this paper, which is called Colonic Goblet Cell Associated Antigen Passages Mediate Physiologic and Beneficial Translocation of Live Gut Bacteria in Pre-weaning Mice. And the first author of this paper is, and I always apologize for mispronunciation of names, Sriram,
And also mispronunciation of the word mispronunciation.
also that, Sriram Udayan, and
Sounds close.
the last author of the paper is Rodney Newberry. And so I really I think this is a cool paper. And being glib about the leaky guts thing at the top, one of the things that I find most fascinating when we run into new papers in immunology is when these paradigms that we have built up in our heads, right? You're sort of taught how immunology works. You get canon, you think about all of these systems. I think about spatial systems a lot. Um, Sometimes these paradigms just kind of don't make sense based on the observations that you see in the world. And yet, these paradigms persist, largely because it comforts us as immunologists to feel like we have a really good handle on everything going around in the immune system, and sometimes it just simply turns out not to be the case. The thing that pops up in my mind is when we were talking about the lymphatics of the brain, for example,
Yeah,
right?
I
Where,
remember this very
yeah,
clearly.
the blood-brain barrier is such an important thing that how could one conceive of the fact that the immune system would be able to monitor that system through a series of lymphatics, which operate I would say broadly, like literally every other lymphatic system,
Yeah.
although into a more tolerogenic immune environment. And so,
I think we did that paper on autoimmunity at one point where
uh,
people were literally just like, hey guys, turns out there are lymphatics in the brain. They operate like every other lymphatic system.
yeah.
Why did we not know this until whatever that was, 2015 or something?
Yeah, no, it took a far too long amount of time to discover that fluid moves through the brain in a meaningful way and that your immune system might be able to monitor that fluid and respond in kind, right? And it's and this was the paradigm that was being challenged at the time, which was the paradigm of immune privilege, right? This idea that there are sites in the body that your immune system simply doesn't see. Um, and it was an important concept because we know, in the real world, when these immune privileged sites stop being privileged, right? When the immune system seems to engage them in a meaningful way, it can have catastrophic outcomes, right? So in the brain, for example, breaches in the blood-brain barrier can lead to things like multiple sclerosis, right? So autoimmune disorders. In the eye, this is something I've come to learn, being married to an ophthalmologist. If, for example, someone has traumatic injury in your left eye, right, what they call an open globe, right? So the eyeball itself is ruptured in some way, right? What happens?
This maybe should have had a trigger warning before you
Yeah,
started
sorry
that,
about that.
but go
It's
ahead.
gross. Actually, funny story about that later, perhaps. But um, if you have a ruptured eye, one of the things that happens is your immune system comes in, as you'd expect, right, and tries to make sure that that infection doesn't get out of control. But a consequence of that is if you don't control that inflammation, in a few weeks, what you find is that your immune system starts attacking the other eye. So people with traumatic eye injury on one side will lose use of their sight on the other side, not because the injury spreads, but because the immune response starts recognizing the eye and responding to eye specific proteins, right? So
Despite
all
the
of
fact that in principle, the immune system shouldn't be able to get to the eye according to dogma.
Right, exactly,
Yeah.
exactly. And so this is these sort of like general observations are that, the immune system doesn't respond in these sites because it simply can't see them. And when it does see them, it's a real problem. You start responding to things in the brain and in the eye and all of these things. But it turns out that none of that is actually true, right?
Well,
It's sort of true
it's
in
not
that.
true in the sense that it is not at all times in all places true. I think
Mhm.
this is again another recurring theme, but this all models are wrong framework. I know we've talked about this before. We build models of things because it is useful to draw boundaries around things. It is useful to categorize. This is true in every scientific domain. I mean, it's true in life that uh, we make models of things. And actually, I have a blog post that I sort of like I have written and never actually published. Um, that's on the relationship of this idea of all models are wrong and this like core Buddhist concept of emptiness, which is like the idea there is that like all concepts are inherently empty. They don't have intrinsic substance. They're like manufactured. And the sort of naive understanding of that is like postmodern or just like nihilism, like everything just like nothing matters, nothing has substance. But it's not quite that. It's that you need to understand when your models apply and when they don't.
Mhm.
And I think, whether it's lymphatics in the brain or it's immune privilege of the eye, like the immune system doesn't operate in the eye the way it operates in your gut or in your skin. It is privileged in some sense. Um, Similarly, like the immune system does have less access to your brain than to other tissues, but it is not everywhere and always true. And the another phrase sort of glib in some sense, but I think holds a profound reality, is like the map is not the territory, right? That is expressing a similar idea of all models are wrong, but some are useful. Like you don't want to go for a hike without a map, but you also can't just look at the map and say you've gone on a hike. Like there's a difference between the model and the constraints that a model puts on your thinking and a way that it allows you to conceptualize a thing and describe a thing and design experiments. And if you treat it too literally, if you treat it too seriously as the actual reality of the situation, you're going to miss stuff. And I think that applies here as well.
Yeah, we got a little more profound and deep than we normally do here on autoimmunity. But no, I think it's important. And that immune privilege conversation is one that we've had, I think, a couple times. And an extension of that today, maybe not the immune privilege, but the idea of a model not working in all places at all times is really the focus of this paper. Right. So
actually, I'll throw another sort of paradigm that has been sort of interestingly tweaked a bunch, which this paper I feel like is an interesting contribution to. So, and that is the concept of the everything microbiome. So we know that there are microorganisms in a lot of places that maybe 30 years ago people would not have thought about. And they're profoundly important and useful to think about in terms of human health. So of course, there's the gut microbiome, incredible density of microbes there, but we've got microbes on like all the surfaces of our skin that are exposed to the outside, in the vaginal tract. There's a microbiome, but then there's been a series of papers talking about microbiomes in places that you like wouldn't really expect. So like the lung microbiome. The lung, generally speaking, we think of as a place that is trying to exclude microorganisms, and that's true. If you have cystic fibrosis, obviously you have a pathology there. Probably the lungs, while they try to exclude many microorganisms, there probably are some that exist there. But then you have things like, there was a paper maybe 12 years ago about the brain microbiome. I feel very confident in saying there is no brain microbiome. I am pretty confident in saying there is no placental microbiome. Like in a typical healthy pregnancy, microbes are going to be excluded from the uterus inside the placenta, right? That like I'm sure there are pathological cases where that's not true. But people sort of report on these because test tubes can be contaminated, even things you get from Eppendorf or whatever. You get a bag of test tubes, like they can have microbial DNA on them. Like microbial DNA is just freaking everywhere. So there have been a lot of reports of microbes in places that there really shouldn't be microbes, and honestly probably are not microbes because of contamination. I feel like this paper, they do a very good job of showing there are microbes where we would, I think 5 years ago, actually before reading this paper, I would have said there are not microbes there. I would have said that same. I'm very confident that there are not microbes in the mesenteric lymph nodes, even of preweaning mice. But I think this paper, I'm fairly convinced by this paper that actually, in this case, actually there are microbes there. And we can get into why.
We should start a show. Are there microbes there? Maybe.
I mean, unfortunately, there's the adage that if there's a question mark in the headline, the answer is always no.
Yeah, right. Right.
There's a lot of this going on right now. Is that legal?
We could break that paradigm.
No.
So
in this one case, yes.
Right, exactly. So let's lay out the paradigm. So the paradigm is that, and we've talked about this over and over again here on autoimmunity, that your immune system's job is not necessarily a fighting force so much as it is a homeostatic force, right?
Peacekeeping.
So it is a peacekeeper, right? And you've got tons of piece to keep with all of those microorganisms that you just mentioned, right? Your skin is covered in normal flora, your gut is coated with normal flora, right? All of these things are not just a part of you, but they help in many cases. They keep out bad bugs, they help aid in digestion, right? These are critical to the functioning of human life, right? And so these are really complex environments that house, millions of species of bacteria. And also sometimes have viruses associated with them, also sometimes have fungi associated with them, right? So we have these ecosystems that live in and around us. And it is your immune system's job to sort of maintain the barrier spaces that differentiate us from them. And so we've talked about this a number of times. Your immune system's job is not to sit in the intestine and control all microbial life and prevent it from existing there. It's more akin to a gardener, right? You're making sure that you're growing things appropriately that are going to be beneficial to you. You're knocking down weeds when you need to. And when there's something really invasive, right? There's a predator in the garden, you need to find a way to drive it out, right?
Side
So,
note, I have what I think is probably a hedgehog
mm,
that is in my neighborhood that has just like destroyed a bunch of my uh perennials. My rhododendron is just like chewed back almost to the ground. Uh and yeah, pain in the butt.
I've learned that squirrels and rabbits in Atlanta will eat literally anything, like including
Yeah.
the wires out of your car. So
Oh yeah,
anyway,
all of my Christmas lights have been destroyed. I didn't take
nice.
them down quickly enough.
Hmm.
Actually, some of them got chewed through like in the dead of winter. But like,
Mm,
yeah, I went to take them down finally a couple weeks ago, and I just like went to the end, and they're just like stripped wires, just
gone.
like just gone.
Nice. Nice. Excellent. So that's your immune system's job, right? Is to
Destroy your Christmas
weed
lights.
out, right? Destroy your Christmas lights. No, weed out bad actors, chase away predators, and cultivate a healthy environment that helps your body function, right? But there's a critical piece of that which this paper picks apart a little bit. All of that discussion is sort of based on this idea that there is a clean and clear barrier that exists between us and them, right? And we've talked about this in the past. Oftentimes, your immune system, certainly an adult immune system, will use the physical location where it interacts with a pathogen to decide just how dangerous, or I said pathogen, I should have said microbe, right?
Right.
To decide just how dangerous that microbe might be. So, for example, if your immune system, let's say you're a dendritic cell, right? And we've talked about dendritic cells as sort of the local assessors and messengers of the lymph node, right? So they're looking around for different microbial life, different proteins, peptides, lipids sometimes, right? Just trying to get a sense of what's going on around them and report back so that the rest of the immune system can respond in kind, right? So those dendritic cells can sit in your intestine. They can like wave their dendritic cell arms, if you can see me on the video, right, into the lumen of the intestine. And if they see a microbe there, not necessarily a problem, right? Microbes are supposed to be there.
Right.
They would be expected to be there. And in fact, when a dendritic cell encounters a microbe in that kind of environment, it might create a tolerogenic response, right? Rather
Exactly.
than
Yep.
trying to be inflammatory, you might actually promote tolerance.
Well,
Now,
and
if
this is related, we've talked before about pathogen associated molecular patterns, which
Mhm.
is this went to a innate immunity lab for grad school, and like this is the sort of one of the buzzwords, PAMPs. And there has been sort of a controversy of like whether those should uh, more accurately be called danger associated molecular patterns. Um, but even that is a little bit misleading because the danger is the combination of the molecule and the location,
Yep.
as you're saying. It is not a problem to encounter LPS in the lumen of your intestine. it is a problem to encounter LPS in your eye. Like there are the context really matters.
The
I
phrase
felt about
"it is a problem to encounter LPS in your eye" is one that will go down in the history books. I like it. Um, but no, you're exactly right, obviously, right? So those same dendritic cells, if you take a version of those dendritic cells that are now not sitting at the junction of the barrier of your intestine, and you drop them down into the dermal layers, right? Or you drop them down deeper into that intestinal tissue where there should not be microbes. And that dendritic cell now encounters those same microbe-associated molecular patterns. Then you have a problem, right? Because that signal now means not only that it encountered that microbe, but it encountered a microbe beyond where the barrier between you and the outside world should be, right? So the presence of that microbe there indicates that there has been a breach of that barrier. And that breach of barrier is a really fundamental concept in immunology, right? We talk about homeostasis between the immune system and the microbial world, surrounding those barrier systems and how the innate system sort of supports those barriers and the microbes support those barriers and all of that. But it assumes this very nice interface between what is us and what is them. And we always assume that when that interface is breached, that it is necessarily pathogenic, right? And the cool thing about this paper is they start to break that concept down, at least in a very narrow window, right?
it.
Where and I didn't know any of this. Reading through their introduction was actually really helpful for me. It turns out there's actually really good reason to think um, that these breaches in barrier systems might happen and might happen specifically in early life, right? So they have a bunch of really helpful references. And I
Yeah.
will admit that I did not go and read most of those references, but just looking at the
Normie.
title, I mean, you are you're looking at the potential for microbial signatures influencing brain development, right? Influencing splenic function, influencing the thymus, right? And we've talked for a long time about how uh, pressure from the outside world on the thymus might have really interesting and
Yeah,
potentially detrimental effects on
for sure.
downstream immune function because that's where T cells grow up and learn how to be T cells, right? So you have all of this potential evidence that suggests that, your microbiome is influencing your body systemically in a way that they do say, some of that could potentially be explained by the dissemination of microbial products.
Yep.
Um, but what was
Or
really,
even the migration of it. So
yeah,
there's actually a quote that I want to read off here because I think it nicely frames the sort of conceptual basis for this project, at least as they're describing it. So it says, current concepts of how gut-resident bacteria mediate effects at distant sites include local activities of bacterial products that affect circulating cellular populations and/or bacterial products produced in the gut lumen diffusing and acting beyond the gut, right? So this is either the microbial products, not the microbes themselves, but the products like LPS, for example, or the cell populations that encounter those in the gut. Both of those can intentionally or potentially move. Then they say, while these models are sufficient to explain many effects of gut-resident bacteria at distant host sites, they appear less adequate to explain distant effects of gut-resident bacteria mediated by products produced in low concentrations and/or distant events mediated by unstable bacteria-derived metabolites, right? So the idea here is that if you've got very, very small concentrations of things, the ability of those things to diffuse to all these distant body sites is very low. And also, again, because context matters, the immune system is not going to respond very quickly to things that are either very low concentration or that degrade quickly. So that's sort of the basis for this, that we know that there are low abundance molecules that have physiological effects at distant sites. So how is that happening?
Yeah, and I would add in two more bits of evidence that they introduce in their introduction, where it probably belongs. One is that they've found microbial DNA in the spleen,
Right.
right? And that's not explainable really by the dissemination of microbial products from the gut, and maybe you could argue that you're getting trafficking in some unclear way from the gut through the innate system, probably through dendritic cells. You could argue that,
Yeah,
but
but that DNA is going to get degraded. Like that seems very
exactly,
hard to. I
it
mean, the
it
explanation I would have had before reading this paper and looking at the evidence would be like, no, this is just contamination of your reagents,
yeah, yeah,
like the
absolutely.
brain microbiome that I talked about before. Like,
Yep.
you know,
Yep. And then the other interesting piece of this is that they cite a few papers, three of them actually, that I'll highlight here, that talk about the long-term effects of inhibiting microbiome contact within a very specific early life window, right? So basically, they cite a paper that shows that if you mess with early life microbiome colonization, you get influences on long-term IgE levels, right? If you mess with that same window, you get a reduction in tolerance generally to bacterial antigen. And if you mess with that window, you get weird effects on long-term natural killer T cell function, right?
hmm,
So those papers suggest that there seems to be a window in early phase life, and this is explainable by other mechanisms too, I think. But there are, because, the colonization rapidly turns over, right? You know this better than I, but you're getting major transitions in bacterial presence there, composition. But it does suggest that there is a really critical window early on in development that has a profound effect on the development of the long-term immune system of the organism into adulthood,
yeah,
right? And that I think is where we really jump into this paper. Are these all one-off things that are sort of weird quirks that we can explain through a bunch of different kind of quirky, weird mechanisms? Or is there something larger going on here that might help explain why all of these things are tied to this early life stage development? And so with that, I think we should jump into this paper.
yeah, for sure. All right, so we've set up the context, and so the first set of experiments. I mean, a huge chunk of this paper is really, I think, trying to nail down this idea that actually this happens and that you get microbial movement specifically from the gut to mesenteric lymph node and to the spleen, right? So the mesenteric lymph node is the lymph node that drains the lamina propria of the gut. So thinking sort of physiologically, the lumen of the gut, the sort of inside of the gut where food is passing through and where the bulk of the microbes in your body are, particularly in the colon, you have a single layer of gut epithelial cells um, that are responsible for a lot of things. There's a few different types of gut epithelial cells. We don't need to go into the whole network of different cell types, but for the purposes of this paper, the one cell type that is important are these cells called goblet cells. If you look on histology, they kind of look like a medieval goblet. They sort of have like a bulbous end and a sort of narrow end. Um, these cells have all kinds of functions. In this case, they're specifically talking about a physiological feature, which I had never heard of before, frankly. They're called colonic goblet cell associated antigen passages, which exactly like they sound like are basically like ways for antigen, uh, molecules associated with microbes that the immune system can recognize to pass, from the lumen across this single cell layer of epithelium into the other side, which broadly we call the lamina propria. So the sort of layer of tissue um, on the basal, so well, I'm wondering how much physiology of gut epithelium to get to, but you have the apical side, which is the side facing the lumen, and you have the basal side, which is the other side. There's all kinds of cool cell biology there, but the lamina propria is where everything else happens. Stuff gets absorbed across the colonic barrier into the lamina propria. That's where most of the immune activity of the gut is housed, right? You have T cells and B cells, dendritic cells, and lots of other immune cells that are hanging out in the lamina propria, trying to block, in an adult, in a normal physiological case, trying to prevent any of this barrier crossing happening. And so these goblet cells, these antigen passages in the goblet cells, these gaps, actually allow antigen to pass through, which is kind of gnarly. In this paper, they're going to be talking primarily about 3 different life stages. So the phenomenon that they're talking about is really happening with mice that are sort of not like brand new, pinkies, not like super new mice, but also before they have weaned, so pre-weaning mice. That are day 17, essentially, day of life 17, so after birth, compared to 21, 21 days, which is when you typically wean mice, so it's 3 weeks. You separate them from mom, and so they're on solid food continually, and then they also have day of life 35, which are basically adult mice. So those are sort of the windows that they're going to look at. And in figure 1, they're trying to say that in these pre-weaning mice, you get spontaneous and physiological, in other words, non-pathological translocation, movement of microbes from the gut to these distal sites, the mesenteric lymph node and the spleen. And so they show this in a couple of different ways. They take lymph nodes, mesenteric lymph nodes and spleens from day 17 mice and day 35 mice, and they show that basically they can find colony forming units of bacteria if they streak those tissues out. They can culture bugs from the spleens and MLNs of the young mice, but not the older mice. They look across a couple of different litters, so this isn't just like a one-off thing. They're sort of seeing this progressively, and then they try to quantitate by doing they do Sanger sequencing. So they PCR up, I think, the 16S gene, and then they do Sanger sequencing of those genes to see what kinds of organisms are doing these trafficking, and they quantify it for a couple of different mice. And there's a different range of bugs. Largely speaking, the bugs that are trafficking are similar to the bugs that you find in the gut, but the proportions are different. So when they look, for example, let me back up for a sec. I'm trying to look at my notes here. So basically, they're saying that it is not that the most abundant bugs are the ones that are trafficking. There are a subset of bugs, even when they're low, they're sort of a small proportion of the gut bugs, they still tend to dominate. And one that they single out that they're going to be using a lot is this Lactobacillus animalis. So a couple of different Lactobacillus species, but in particular, this Lactobacillus animalis is one of the ones they see frequently in the mesenteric lymph node and spleen, even when it is not a particularly abundant component of the gut microbiome.
Yeah, which begs the question, right? What is it specific to those lactobacilli species that is allowing them to seemingly take advantage of whatever it is that the phenomena is that we're going to lay out downstream?
Yeah, exactly. And so there's sort of like, at the end of this figure, an argument you could still make is that this is some contamination happening. Even though they don't seem to get any from the older mice, so I wouldn't say this is contamination of like the test tubes and the pipettes and things. But you could imagine that doing surgery, extracting the mesenteric lymph node of a 17-year-old or 17-day-old pup versus a 35-day-old like full-grown mouse, you can imagine it's much harder to isolate just the mesenteric lymph node and spleen from these tiny little pups. You could imagine it might be easier to cross-contaminate with other sources. But if that was the case, you would expect it to be sort of stochastic and reflect the contents of the gut. So that this is in part an argument like, well, this probably isn't random contamination. There still might be other explanations, but the argument they want to make with this is that the fact that it is specific to a small number of bugs and is not dependent on the colonic proportion of these bugs suggests that there's some kind of active physiologic process that is facilitating the movement of certain kinds of bugs and not others.
Yeah, and I will point out, for the rest of this paper, we're going to be talking about Animalis, right? LL
Yeah.
Animalis for the most part. It's important to focus when you've got a story down on one thing. I mean, one of the things that I think is just a function of how these kinds of stories develop is, we're going to spend the last bit of this paper trying to figure out exactly what that one particular bug does.
Right.
But I think the broader context here, right, is that there are a number of things that can take advantage of this system and potentially interact in interesting ways with our immune system. And I'll point out later on in the paper where I think we're maybe getting too much in the weeds, tracking down one specific species and what it's doing and all of that. But I think the broader take home from figure one is that this can happen, right? You can get translocation actively from the gut into that extraluminal space, which is pretty fascinating.
Yeah, which is very unexpected, very unexpected. I think the fact that we don't see it at all in the adult mice is instructive in the sense that, again, like it's not that the sort of typical model of barrier function is wrong. It's just that it is context dependent.
Yeah.
Once you have a developed immune system, once you are weaned and going out into the world, it makes sense to have a little bit more control because you're going to be eating lots of stuff. Lots of stuff has potential entry points. You can be in really bad shape if you encounter pathogens. When you're primarily consuming mother's breast milk, the opportunities for infection via the oral route are reduced. They're not gone, but it's reduced. The fact that this is not just every bug can just make its way to the lymph node, there's some kind of control over this process. All of those things suggest that there's some benefit to, or some sort of evolved and, obviously we don't want to impart intention to evolution, but colloquially, some intentionality behind this movement. The other thing I think is notable here is that this is not happening in every pup.
Yeah.
Which itself is kind of fascinating because not every pup is like, it's not as if this is preventing some kind of, horrible outcome. Because the pups that aren't seeing these translocations, you could argue like maybe it's sort of time dependent and it happens at some point, but they're not catching the right window for every single pup or something. Who knows? But regardless, it seems that this is not a thing that is happening in all pups all the time, but it can happen and it's not pathologic when it does happen.
Yeah, and I would say, because I've vaccinated a lot of Black 6 and infected a lot of Black 6 animals that are being used here, right? These pups will all become quite immunologically similar.
Right.
Right. So, the stochasticity that we're seeing here in the early translocation on the back end does not seem to have a huge, meaningful impact on the long term in the ways that we are assessing, right? In the ways that we are assessing. That said, like you said, it is not clear at what time point we're catching this, whether we shifted the window by 6 days, maybe, each individual animal has a spike and then that spike closes and we wind up in the same. But it is interesting, you're right, that there is more dynamic range in what we're seeing in these animals.
Well, and to your point too about the similarity of these mice, think back to the paper we did a couple episodes ago on the rewilded mice.
Yeah.
Right. So these are mice in confined facilities, also genetically inbred. They're using a single strain. And so we we are seeing this phenomenon in this particular mouse in physiological conditions
Mhm.
out in the world when exposed to a wider range of microbes in a population with a lot more genetic diversity. We don't know how these things might affect um, responses to infection. There's so much variation in the way that organisms respond to infection that a lot of it we chalk up to different experiences, a lot of it we chalk up to genetic differences. This is potentially one more thing that is altering the immune landscape onto which a future pathogen or a future insult that could lead to autoimmunity could land, right?
Yeah, and they
And it's
and
just previously unobserved
yeah,
variation.
and they dig into that a little bit, that possibility a little bit in figure 4.
Yeah, exactly. All right. So figure 1, we're seeing this translocation. Figure 2, we're going to start to try to dig in to understand. So here they're turning to a couple of different model systems that are variations on themes that we've talked about before. So one is they have a Cree mouse where they can essentially knock out those goblet cells we were talking about that form um, these potential pathogens. And they're now, for most of the rest of the paper, they're now colonizing these animals with this strain of Lactobacillus animalis to essentially like boost the frequency with which this is happening. And because they can track this bug, it's got a fluorescent reporter, et cetera. And it's sort of easier to culture and quantitate. So basically, so they look in these animals where they either have or don't have goblet cells after an induction with Cree, and they show that the amount of Animalis colonizing the gut is not different between the mice with goblet cells and the ones without them, but that when you don't have goblet cells, you basically lose all of that translocation that you're seeing to both mesenteric lymph node and spleen. So the concentration of the bug in the gut is the same, but translocation stops happening. They also show that if you treat mice with EGF, which they explain this because mother's breast milk has EGF in it. High concentrations of EGF are suppressing these goblet cell antigen passages, and so they're sort of treating with EGF. They see a reduction in the goblet cell passages, and again, they see no change in the amount of bugs in the gut, but you see substantially decreased translocation of these microbes into external tissues. They still have some, so it's not like a full reduction. I'm not
Which
a huge
is
fan of this model because EGF like does so much stuff that it seems weird, but we can leave that aside for now.
Yeah, well, I'm going to have a bigger problem with the next model,
Yeah, later
so
on.
go ahead.
No, I think it matters more later. In this figure, the broader point is there are different ways of blocking this translocation. And to me, particularly the Cree mouse, to me, is the best evidence that this is not accidental contamination.
Yeah.
Because these are the same age mice, so any of those problems that you might have had with like these surgeries being tricky or whatever, cross-contamination with the gut, any problems that you might have there, the fact that we're knocking out a single cell type basically puts those to bed. You knock out this cell type, you don't get that translocation, and then you don't see this dissemination. So I thought that part was pretty convincing, at least. They also show in this figure that was this a knockout of dendritic cells? I'm trying to remember. Oh no,
Well,
the quadrosome.
yeah, so
So
they use FTY 720, which is interesting, right? Because FTY 720 is an inhibitor of the S1P
right,
receptor pathway,
right.
right? And so this is a pathway that is frequently used for you usually use this drug in the context of locking um, locking immune systems into their respective secondary lymphoid organs, which is
Right.
interesting, right? So a B cell and a T cell, if they're looking to leave a lymph node and travel to another lymph node, and I can never remember which one. They either, I think that they upregulate S1P receptor, right? And
Mhm.
S1P is being produced in the medulla of that lymph node. So lymphocytes looking to leave immunologic secondary lymphoid tissue, they need to make use of this pathway.
Right.
And if you introduce FTY720, it's a really potent inhibitor of that pathway. So locking or adding FTY720 is generally used to prevent B cells and T cells from leaving secondary lymphoid tissue. I am not familiar, and that doesn't mean that it is not correctly used here. I'm not familiar with the use of FTY720 as a general
Yeah, they say in the text that
eliminator.
this is blocking DC migration
Yeah,
uh
which
to the
which could
lymph
well
node
be.
from the lamina propria.
It could well be. I'm not familiar with that literature. Could well be, but it is interesting. I think the FTY 720 is actually a better model if it is working the way that they say that it is, and
Yeah.
I have to trust that it is. I do believe that that blockade of dendritic cell trafficking would be a better representative model than the addition of EGF.
Yeah.
Um, which is why it is confusing then that we move into the collagenate loaded liposomes. Um, so why don't you introduce that, and then I will tell you all the things that I hate about them.
Fair. So I think the point that they're trying to make broadly with this series of experiments, maybe it's easier to set that up,
Mhm.
is that they are claiming that you are basically getting uh microbes that are being taken up but not killed by cells, by immune cells like DCs, like macrophages, and that those cells are then the ones that are shuttling the microbes intact to the mesenteric lymph node in the spleen, right? So if you imagine a situation, it's not that microbes are getting into the lamina propria and then diffusing with the lymph up to the MLN and eventually entering the blood and going to the spleen, right? So they're not saying that these microbes are disseminating systemically like you would have in sepsis. What they're trying to claim is that you have immune cells that are engulfing these bugs and then bringing them to their respective locations. And so the EGF is sort of trying to block the movement from the gut into the lamina propria through these goblet cell associated passages. The FTY720 is then saying we're going to block the movement of the cells themselves that have engulfed them. We're blocking their movement. The cladrosome, so my understanding of cladrosome is that basically the phagocytes pick up these liposomes and then it kills them, or it uh yeah, it basically wipes them out.
Yes.
And then I'll just say that, and then they see a reduction again in those. And then the last thing they do before you get into your criticism of cladrosome is
Mhm.
they treat with gentamicin, which is a antimicrobial that won't cross cell membranes. And so they show that the gentamicin, if you just treat bugs in culture with gentamicin, it readily kills Lactobacillus animalis. But if you treat the mice with it, the cells, if they've engulfed the bacteria, the gentamicin isn't going to get to those bacteria in the cells, and they see that you still get translocation in the presence of gentamicin, which is another piece of evidence they claim that you have bugs inside of immune cells that are responsible for the trafficking. So there's like three different pieces of evidence, all trying to point to the same concept. So
Yeah. Yeah, and I think that it's fairly strong. My worry with the collagenate loaded liposomes is that you are absolutely correct. They're these little, right, liposomes that phagocytic cells pick up, and it wipes them out. And it does so in a spectacularly inflammatory way.
Mm.
So these have been used routinely in lymph node biology to assess, and I, to be honest with you, I'm not as up to date on some of those studies in
Yeah,
the
you did this
last
stuff
like
in grad
5 years.
school,
Yeah, this was grad school stuff. But at the time, people were looking to understand the function of all of these different macrophage populations in the lymph node.
right?
And one of the things that you did routinely was you would inject these collagenate loaded liposomes and they would diffuse through the lymph and they'd be picked up by subcapsular sinus macrophages and medullary macrophages and all these different populations that are phagocytic in nature. And then you could show what that system would look like if you just didn't have macrophages in it. Well, you did two things when you did that. The first thing you did was you depleted the macrophages. The second thing you did was caused all of those macrophages to freak out simultaneously and release a whole bunch of inflammatory cytokines.
Yeah,
So the combination of destroying the barrier that those macrophages provided, right? Because you're leaving physical holes in the
yeah,
barrier when you're doing this, right? The combination of that and also the fact that you are releasing all of these pro-inflammatory cytokines meant that you were fundamentally changing the environment that you were evaluating.
right.
And what we are talking about here is a potentially very delicate system where you've got a tolerogenic anti-inflammatory system with this sort of active passage of bacteria. That
Yeah,
doesn't mean that it's going to be dumb, right? It doesn't
right.
mean that in all cases it's just going to keep shuttling bacteria in exactly the same way, right? You could imagine a situation where if you start activating macrophages in this local environment, you are fundamentally changing the properties of this barrier system, and you are actually preventing the possibility of this nice
Yeah,
kumbaya
for
tolerogenic kind
sure.
of thing happening, right? Now, the FTY 720 is the intriguing part to me, right? Because if you are truly just blocking dendritic cell migration from site to site, and it's
Yeah,
having this profound an effect, I mean, you're going from total translocation to nothing,
to nothing.
right?
Yeah, absolutely.
That is mechanism, right? You could have followed that up and used a dendritic cell knockout animal, for
Right.
example, right? And if you could show that just by eliminating in a non-inflammatory way the shuttling dendritic cells, then you could have just walked away from the mechanism,
Yeah.
right?
No,
Now,
totally. I
because
think that's right. And I think, yeah, there are genetic ways of dealing with DCs. We could have done like a DTR kind of system we've talked about in the past to eliminate them so that's not like, not eliminating DCs from the get, but like clearing them out
yep.
just before you measure. There's all kinds of things that you could potentially do. My read on this figure was that they care less about proving that this is the specific because you could
Sure.
always imagine like maybe this isn't a CD11C positive cell that's doing the trafficking, or it's both those and
Yeah.
other kinds of macrophages, or you get, so I feel like my take on this was that they were trying to just show from a couple of different angles the collagenate, yeah, like maybe it's a fucked up system, but it is at least not disproving
Yeah.
their point.
No,
So
it certainly
I
is.
gave it a
And
pass.
the gentamicin, again, a little tricky because both of these organs are filter organs,
Yeah,
right?
absolutely.
And the purpose of those organs is if you have passive flow of something like a bacteria that's hanging out in the lymph, it's
Right.
not just going to continue to exist in
Exactly.
solution, right? You're going to have macrophages that are designed to pick all of those things up. So, like, it's
Right.
not a perfect figure, but they are starting to get at mechanism, and I am very, very intrigued at the FTY 720 data in
Yeah.
particular.
Yeah, that's
Yep.
cool.
Yep.
All right. So that's figure two. Um, figure three, they want to say now that this is not, again, this is not pathological, right? So they've shown, I think in figure one and two, they've pretty convincingly shown you get this translocation. Not all the time, but you get translocation. There's it looks like it is active in some sense. In other words, it looks like it is not just microbes from the gut are just like flowing willy-nilly through the lymph node and into the blood. There's some kind of cellular active process that is occurring both because we're selecting certain bugs to do it, and there are these genetic and chemical tools that kind of demonstrate that there is mechanism, there's some function behind this other than just stochasticity. Now they want to say that this is not inflammatory. And here's where they use the EGF as part of it. But to me, this is problematic because I'm not sure what the anti-inflammatory properties of EGF are or the inflammatory properties of it are. But I know that EGF is not passive with respect to the immune system. So you have this system where you are treating with a drug that is both affecting these antigen passages and also modulating the physiology of the system. And for all of these experiments, I was really curious, why would you not go back to your original system in figure 1 and just pick the mice where you see translocation and compare them to the same mice where you don't see translocation without any chemical or genetic manipulation? Like they see it's a stochastic process. You don't always have it. So just compare inflammation in the animals that do have it and the animals that don't. I don't understand why they didn't do that. But regardless,
I think it's because of the sorting, right? You can't streak out and find which animals have it and then sort the cells that they're trying to sort in order to get these transcriptomic
yeah, but
signatures.
these aren't survival extractions that they're doing, right?
Yeah.
They're like, you could take the cells, you could sort them,
So you sort all you sort all
and
of
then you
the
go back and you say
MHC2.
which ones had the translocation and which ones didn't.
Fair enoff, and just store them in RLT and do the downstream transcriptomics.
Exactly.
Yeah, I
mean,
can see that.
One argument against that is that you could say, well, maybe these mice had translocation that happened right before you extracted, and you can't tell the difference between ones that had it just after. So,
yeah,
there are arguments against it, but the EGF thing, well, it doesn't matter. What they show basically, so they have the animals, they've given them this modified L animalis, so they're sort of ramping up the amount of translocation they see, and then they compare ones that are treated with EGF, which have reduced translocation. It's not gone. This was the treatment that had the least profound impact on translocation itself, which is right. Well, we can argue about whether that's the most appropriate one to compare, also the physiological effects of EGF. But in any case, they do some bulk RNA sequencing, and let's see, they're looking at the MHC2 positive, CD45 positive, they're gated on CD45 positive. So they're basically looking at antigen presenting cells that have MHC class 2, which is a broad array of cell types, but they're doing bulk RNA sequencing.
But not B cells. So they're intentionally excluding B cells from that mix, which that makes sense.
And basically, so they show a bunch of genes that are differentially expressed. I sort of scanned that list and nothing jumped out at me, but the bulk of this figure is panels C through J, where they show a bunch of different inflammatory cytokines and inflammation-associated genes. And basically, they show there's no difference in any of their treatments. So whether or not they add Lactobacillus animalis, whether or not they treat with EGF, basically the levels of all of these inflammatory markers are identical. Where did they extract these cells from? I forgot about that.
So everything that you're looking at in C through J is systemic.
Well, they got the cells from somewhere. I mean, this is the same cells they used in B, right? Or no? Those are ELISAs in the blood.
Yeah, these are blood ELISAs and neutrophil counts, and exactly.
Okay,
so what you're looking at is there a systemic inflammatory signature associated with this?
Yeah, and there isn't. And if you look at the APC RNA-seq, they only do 2 animals per condition. So who knows what that means, but basically they don't see any inflammatory genes being the most differentially expressed genes. It's a bunch of other stuff. Sure. So again, the sort of hard to prove a negative. Yeah. And also there's not a clear like, oh my God, this is a terrible thing that's happening.
Right.
At least for the translocation of this particular bug.
Of this particular bug, right? And this
Yeah.
goes back to the initial question of, do we need to focus on this one thing? Of course, we want to know more about this one bug that seems
Yeah.
to have high levels of translocation. Whether this is true of everything that would cross that barrier, hard to say. And like you said, Kevin, we're unfortunately influencing the system a fair amount by adding in that EGF treatment. So
Yeah.
not super clear here, but getting towards, I think, the idea. And I think all of the other data suggests that this
Right.
is not deeply pathogenic, this process.
Right. I mean, the
right?
fact that it's happening in like,
Yeah.
you know, a not a majority of animals, but a large number of them,
Yeah.
this is occurring, particularly when you feed them this L. animalis, suggests that this is not broadly pathogenic. Like,
Right.
If you put it into a very inflammatory system, would you get potentially more? I think they would argue it'd be more tolerogenic, so you might actually get like less autoimmunity and stuff. And there's, but to the extent that they can show this doesn't seem to be pathological, it doesn't seem to be inflammatory at least.
Yep.
And then so in figure 4, they want to suggest potential benefits. And I'm a little ambivalent about this figure because on the one hand, like reviewers are going to ask for this figure, right? Reviewers want like to show impact. They're going to want to see something like this.
It's a Nature microbiology paper after all.
Yeah. And so fine, like I understand why they did it, but on the other hand, this mechanism that they show, it feels like beside the point almost. Like the tolerogenic stuff, well, let's get into what they show. So what they do some genomic sequencing of this L. animalis strain that they've been using this whole time. And basically they show a couple of they use this software called Anti-Smash, which is looking for secondary metabolites or genes involved in secondary metabolite synthesis. Secondary metabolites are like antibiotics are one class of secondary metabolites, but it includes a bunch of other stuff that's sort of like, not crucial molecules being produced, but other kinds of things that humans don't generally produce from our genomes. And they find a cluster of genes that seems to be associated with antibiotic production, or is similar to a class of antibiotics. They show that if you take spent broth from culturing L. animalis, that broth has antimicrobial activity against an Enterococcus species, or maybe E. coli, a pathogenic form of E. coli. If you treat with proteinase K, the activity goes away, so that suggests that it's a protein antibiotic or antimicrobial. And then they show in what I think is the least convincing experiments in this paper. It's not to say that it's not convincing, but it's just kind of like, it's a little ho-hum. But basically, if you infect these animals, these pre-weaning animals, with a pathogenic strain of E. coli, they have a lot of bad effects. If you also include L. animalis, you don't get as much of these effects, but you can also delete this secondary metabolite gene, and you don't prevent it. So it's not like just a sort of niche occupation. And you get reduced effects when you treat with EGF, or at least you get more pathology when treated with EGF, suggesting that the translocation piece is an important part of the protection.
Right. Assuming that EGF is not having an influence over the larger system.
Yeah, absolutely. Right. Which I think, to me, like the production of a protein antibiotic, it's sort of, the mechanism doesn't make sense. If the translocation is protective, why would you need that antibiotic produced in the mesenteric lymph node? Like the pathogenic E. coli is in the lumen of the gut primarily. I mean, it's causing inflammation. It might get into the lamina propria, but like
Well, and the larger piece of that, I think, is that you've argued up until this point that it's cellular mediated, that
Right. Exactly.
trafficking is inside of a dendritic cell or
Right.
inside of a monocyte, right? That was the point of
Yeah.
the gentamicin experiment
Exactly.
earlier. So you're arguing that the production of antibiotics inside those trafficking cells is somehow protecting you from
Right.
E. coli in the lumen of the gut. And
Right.
I don't get it. I
Yeah.
don't get
No, I
it.
don't either.
That doesn't mean that there is not a protective effect here that I don't
Sure.
understand.
Sure. It could be
But
there are multiple protective effects. Or,
right,
like I think one argument you could make is like, well, this is showing that, the immune system is good at promoting the growth of bugs that have beneficial effects.
absolutely.
Maybe one of the ways that you promote their growth is you take them to the nice cozy environment of a mesosteric lymph node or something, or you're tolerizing, right? You're
Sure.
training the immune system to not attack but like I don't see how there's a mechanism that the immune system could know about the production of the antibiotic. So then you're just saying like, well, maybe we're just helping all lactobacilli. It's a very it's not a very clear um story. It's not a clear part of the story. I
Yeah.
think the strength of this paper is showing that there's this phenomenon of microbes being in a place where you wouldn't expect them,
Mhm.
and they're getting there with the help of the immune system,
Mhm.
or at least that's the claim.
And it seems
Yeah,
right.
this figure feels like it doesn't add to that story.
Yeah, I mean,
Like,
you
oh,
know
and
that
also
I'm
this bug is interesting, but
right. You know that I'm big into making a cartoon, right? And it's
yes,
because I like to think about and then reflect to the people that I'm talking to the physical spaces in which I am thinking about these sorts of
well, and
questions.
also it makes you clearly articulate your model,
Right. Right. I would
and
struggle, I think, to draw a cartoon of what I believed was happening here
right,
based on this paper. And maybe they have one, right? And it is,
yeah,
they're just not there yet, and that model will become clear. But from the data presented in this paper, I would have a hard time drawing that cartoon. I would not have a hard time drawing the fact that these bugs are clearly and selectively being passaged through these interfaces. I think that
right,
is fascinating, potentially impactful, and like you were talking about earlier, a really solid exception to this paradigm that we have built for ourselves that these barrier systems must be intact at all times, or else we get widespread inflammation and sepsis, right?
exactly, totally, totally agree with that. But all in all, I mean, I think it's a good paper.
Yeah.
I think it's a neat phenomenon.
Mhm.
I think there's a lot of work to be done to sort of measure the impact. I'm really fascinated. I would love to see more exploration of the differences between the mice where you see this and the mice where you don't.
Yeah.
Um, it's a bit hard because you got to sack the animal to see the bug in the lymph nodes, or in the spleen.
Yeah.
So
Even
if there
just
was a
to
way that you could, there's all these systems where you can do like almost pulse chase kind of things where you can like if they enter a certain area, like something gets activated and then they're tagged and then so like I feel like there are potential ways to explore that more deeply, but it would be a lot harder, um, but potentially more impactful. I also have a few nitpicks that I want to run down quickly before we end.
Mhm.
If you're
Yeah,
okay ending on
let's
that
do
note.
it.
Do you have
Yeah.
any other broad senses here?
No, I like I said, I think I think it's a good paper. I think it starts to look at an important mechanism. It would be really, I think, illuminating to understand what the kinetics of this actually look like.
Yeah,
We've picked three windows here, really one window
right.
at the end of it. And to your point about stochasticity, I think that I would really be interested to know exactly when this is happening in all of these animals, if it's happening in all of these animals.
Right.
And like you said, if it's not happening in all of these animals, what is the differential impact?
Yeah.
But And of course, then you get into, the multiple strains that we're talking about. We've interrogated one here.
Yeah.
Yeah, it could be that we have differential effects of this depending on, what the primary colonization composition looks like. So, really cool work. Lots of open questions, I think, to
Yeah.
follow up on, and I'm sure that they are working on that.
Yeah. All right. I'm just gonna share one of the nitpicks.
Mhm.
This is a statistical nitpick, and I think you are sometimes guilty of this as well. I
Probably.
just want to state for the record,
I'm guilty of most statistical problems.
if you are using null hypothesis testing, which is like basically anything that generates a p-value, and you are saying that p less than 0.05 or whatever threshold you set is the standard by which you are saying that you are refuting the null hypothesis, do not then say that something is trending toward anything if you don't reach your p-value. Just don't say it. If your p-value, if like there's problems with null hypothesis significance testing, there's problems with p less than 0.05 as the threshold. There's endless debate on the internet. You can go read it if you care about. But I don't want to get into any of that. What I want to say is if you are going to say that p less than 0.05 means that you can reject the null hypothesis, a p greater than 0.05 means that you cannot. And if you cannot reject the null hypothesis, then saying it's trending towards anything is a meaningless phrase. It's just meaningless. So please don't do that. And they do that at least 3 times in this paper. And I encounter it and it makes me want to tear my hair out. It's just, don't do it. Please don't do it.
Can we, as a community, agree not to set that hard threshold and say that perhaps we'll treat p-values on a linear scale and say
Absolutely.
that we can, yeah.
100%. If you are going to make the argument that some combination of effect size and statistical testing, you are going to assess that holistically, then if you want to make a principled argument for that, you can. I don't think it's very easy to make a principled argument for that, but if that's what you want to do, I use p-values too. I'm not going to go against it because that's tradition, or that's
Tradition.
that's people do that, and reviewers want to see it, and journal editors want to see it, and fine. Okay. If you want to make the principled argument for something different, I'm, Godspeed. But if you're just going to say p less than 0.05 is a threshold, and anything that's less than 0.05 is significant, and you're using that as evidence, then you cannot say, if it is greater than 0.05, you cannot say anything about the trend of that thing. There's another place where they say things are enriched, they do FDR, they do false discovery rate correction.
I saw that.
Which is a good thing to do if you're testing lots of hypotheses.
Yeah.
You do FDR correction, that's important. But then they say oh, we see all these things that are enriched but failed to pass FDR correction. And it's no, they're not enriched. If they failed to pass FDR correction, you don't know what they are. You don't know if they are enriched or depleted because you can't rule out the null hypothesis.
Right.
Just period.
Yeah. I did. I saw that, and I knew that was what I thought you were going to bring up, actually, as you entered into this statistical rant.
I mean, listen, when I was in grad school, I didn't pay a lot of attention to statistics.
Because they suck.
They do suck. I don't disagree with that. I think that there are a lot of things that we could do as a field to improve our statistical rigor. And I mean, honestly, in a paper like this, in many cases, many of the phenomenon they're looking at, you don't need a statistical test to see that one column has a bunch of things and the other column has none. They do a statistical test, but it's it's there or it's not there.
Yeah. Yeah.
And I think those are the kinds of experiments we should be striving for. We can't always do those, especially when we're dealing in, diverse genetics, when we're dealing with human subjects, like stats have their place. But if you're going to use statistics, by God, use them correctly, please. And don't self-contradict. That's the big thing. Like, I'm not one to say what is correct use of statistics. I'm not a statistician. Frankly, I'm sure that I have used them in ways that statisticians, professional statisticians, would blanch at. But I think I can say that in all cases, I am consistent in the claims that I am making about the statistics that I am using. And that I think is important.
All right. Well, now, as a listener base, you have full license to go back through all of Kevin Bonham's previously published work where he was the first author and look for that consistency. I think it'd be close. I think it'd be close.
Go on Google Scholar. Take a look. Everything's out there.
Yeah.
And if something's not open access, let me know. I will send you a PDF.
Right. Excellent. Well, another statistical rant brought to you by Audio Immunity, where we have angered our local person that deals with statistics generally more than I do. So,
yeah.
All right. That was fun. I have to say, for having one go at this and no possibility of takes, I think we stayed pretty on course for this one. Now, it is concerning that we, having stayed on course, still took an hour and 20 minutes to review this paper. So at some point we should consider our own long-windedness. But for now, this has been Audio Immunity. That's all I've got. No, you can find us on the web. I got it. You can find us on the web at audioimmunity.org. That's AUDIOMMUNITY.org. Please like and subscribe to this podcast. Leave us ratings on iTunes and support us on Patreon at patreon.com/audioimmunity. And the music at the beginning and the end of this episode is written and composed by Rachel Reinick. Thank you all.
All right.
Sometimes